Hello, my name is Christine Kim, and I'll be presenting my project, PdG ADDUCTS in Culture Cells from Secondhand Electronic Cigarette Smoke. Tobacco cigarettes go through incomplete combustion, which results in carbon monoxide, which is a poisonous gas that, once combined with hemoglobin, blocks blood's ability to properly carry oxygen to body cells. Electronic cigarettes were first thought to be safe because it uses a heated organic solution made by harmless solvents such as isopropylene glycol and vegetable glycerin instead of going through incomplete combustion of tobacco cigarettes. As a result, it has been promoted as safe and used as a gateway to reduce tobacco smoking. Because electronic cigarettes were promoted as safe, its popularity has risen greatly, with currently 3.6 million students of middle and high schools using electronic cigarettes. According to a survey from 2017 to 2019, 8th graders who smoke electronic cigarettes rose from 4% to 9%, in 10th graders 8% to 20%, and in 12th graders 11% to 25%. Electronic cigarettes have been very popular among teenagers because of their accessibility, targeted advertisements, various flavors, and the belief that they are safer than traditional tobacco cigarettes. Despite being promoted as safe, nicotine from electronic cigarettes can be metabolized into formaldehyde, which induces aldehyde adducts in vitro. DNA adducts are covalent interactions of carcinogenic chemicals and DNA. 80% of inhaled electronic cigarette smoke is metabolized into nicotine, which is excreted into the bloodstream. Although nicotine is believed to be non-carcinogenic, about 10% is metabolized into nitrosamines in vivo. Nitrosamines are metabolized into N-nitrosonor-nicotine, NNN, and nicotine-derived nitrosamine ketone, NNK. NNK can further metabolize into methyl dioxyl hydroxide, MDOH, pyridobutyl derivatives, PBDS, and formaldehyde, a type of aldehyde. Formaldehyde specifically induces DNA damage as aldehyde adducts in vitro. In my project, I focused on formaldehyde's effects on UROtsa cells by looking for the presence of DNA adducts, since DNA adducts levels in normal cells serve as a biomarker for significant exposure to carcinogens. Specifically, I'll be looking at PdG ADDUCTS, which are DNA adducts from electronic cigarettes that are known to form in the lungs, heart, and bladder of electronic cigarette-exposed mice. In my research, I will use PdG antibodies to determine if aldehyde adducts formed in UROtsa cells exposed to nicotine from secondhand electronic cigarette smoke. My hypothesis is that aldehyde adducts will form with increasing concentration in correspondence to the length of secondhand exposure. To test my hypothesis, I needed electronic cigarette air samples. To do this, I pipetted 1 mm PBS solution into 18 Eppendorf tubes, which was then divided into 3 groups of 6 tubes. PBS solution was used because it is hypotonic with human cells, which will be important for the next step. Each group of 6 was placed in 3 rooms of frequent electronic cigarette smokers. Lastly, the tubes were left open, and after 2 weeks, 2 tubes were closed. The next 2 were closed after 1 month, and the last 2 were closed after 1.5 months. The longer the tubes were left open, the more nicotine was absorbed from the air as a form of secondhand cigarette smoke. To mimic the absorbance of electronic cigarette smoke, I use UROtsa cells, which is commonly used to study the effects of metals and toxic substances due to its sterility. My mentor exposed the culture cells to the PBS solution for an hour in order for the cells to absorb the nicotine present in the solution at New York University Environmental Labs. Any other potential hazardous substances in the PBS will not be able to affect the cells since 1 hour is too short to cause any effects, whereas nicotine is easily absorbed into cells. A control of pure PBS solution was used to ensure that the results are due to secondhand smoking and not from any issues during the DNA extraction and analysis steps. In order to see if any adducts were formed from the PBS solution exposure to the cells, I needed to first extract the DNA. To do this, I used the phenylchloroform method. First, I lysed the cells with a lysis buffer. Then I pipetted equal parts phenol to the Eppendorf tubes the cells were in. The phenol broke down the protein in the lysed cell supernatant, and the protein was left in the phenol, which was then removed with a pipette. This step was repeated 3 times in order to ensure all of the protein has been removed. Next, equal parts chloroform was pipetted in order to denature the protein that may have been missed during the phenol step, and then the chloroform was removed with a pipette. This step was done 2 times. Next, RNase was added to break down the RNA that would interfere with my DNA concentration calculations. The DNA was then precipitated by using ethanol and placed in a negative 80 degrees Celsius scientific freezer. Lastly, the ethanol was removed and 50 microliters of 1-TE was pipetted onto the DNA. This was all done in a fume hood with proper safety measures, as phenol and chloroform are dangerous and carcinogenic substances. In order to test for PdG ADDUCTS, 600 nanograms of DNA was needed, which meant I needed to measure the concentration of each sample. To do this, I used a spectrometry machine which analyzes your sample of DNA concentration, which the results are shown in the red circle in this picture. With the concentration of each sample, I calculated how many microliters of each sample I needed to achieve 600 nanograms of DNA. I pipetted 600 nanograms of DNA and positive controls into nitrocellulose membrane and used a vacuum to leave only DNA on the membrane and remove the TE solution it was in. The positive control was used to ensure that there was no problem with the blotting procedure. The tool I used for this step is shown in this picture. The vacuum sucks out the TE solution and leaves the DNA on the membrane. After this step, the membrane was carefully put under a UV lamp in order to bind DNA to the membrane to ensure that the DNA does not fall off. The membrane was then placed in a small box with PdG antibody solution poured over it and rocked for an hour. If there were any adduct formed, the PdG antibody would have bonded with it. Next, the solution was poured off and the membrane was washed using a wash buffer to ensure that the PdG antibody was not left on the part of the membrane without any DNA. A second antibody was poured to allow the stain to bind to the DNA if there were any PdG adducts present. Lastly, the stain Q dot was poured over and the membrane was left to rock for an hour and a half. If there were any adducts formed, the stain would stick onto the second antibody. In summary, if there were any adducts formed, the layer would be in order would be membrane, DNA, adduct, PdG antibody, second antibody, and then the Q dot stain. The image of the adduct formation was taken on the BioRad Gel Doc XR Plus with Image Lab software and machine, as shown in the image on the right. Adduct formation can be seen because of the Q dot stain. After taking an image with the BioRad machine, the positive controls can be seen with a dark line indicating the presence of PdG adducts. Slots of the nicotine-exposed DNA show the same shade of gray as the negative control. Since the shade of gray of my sample is the same shade as the negative control, it is an indication of no adduct formation. This means that it is concluded that 1.5 months of secondhand exposure is too short to cause any adduct formation once the nicotine is metabolized. Since my project encompasses secondhand electronic cigarette smoke, the effects are much more toned down than the effects of first-hand electronic cigarette smoke. However, the method I used may not have been sensitive enough to detect all aldehyde adducts, hence my results. It cannot be confidently concluded that 1.5 months exposure to electronic cigarette smoke has exactly no damage in the DNA. The conclusion of my research is that 1.5-month exposure to secondhand electronic cigarette smoke has little to none aldehyde adduct formation in the DNA of UROtsa cells. However, as stated above, this conclusion may be wrong as my methods may not have been sensitive enough. In addition, the concentration of absorbed nicotine may have been too little, which can be solved by lengthened absorbent time. By doing more specific experiment, my research can be solidified to ensure that there is indeed no aldehyde adduct formation. Nonetheless, because research in this field is new, and there is not a lot of research done on this topic, my research can be used as a basis for new research on genetic secondhand effects of electronic cigarette smoke. In my future work, I hope to expose the UROtsa cells longer in order to maximize the nicotine absorbance. I would also like to collect secondhand smoke for a longer period of time to increase the concentration that can be collected. Lastly, I would like to use a more sensitive method in order to maximize my results and to ensure if there has been any adduct formation or not. These are my references.